thank you to Alex and Phoebe for this wonderful invitation. I'm thrilled to be talking about this research today. I wanted to first definitely acknowledge my colleagues, friends, co-authors, Julie Lesnick and John Speth, that helped with this research. But I wanted to start out with kind of a who am I? I am someone who's really interested in various questions of environment-human interactions. So I work on projects in paleoanthropology, early, around 4 million years ago, usually at Alia Bay, when the environment is shaping early humans, and then in California bioarchaeology at a time when rising populations are impacting the environment around them. And so their hunting choices are kind of shaping the local ecology. And then in forensic anthropology, when we are trying to identify unidentified individuals and can use the fact that the environment will imprint life history information into biological tissues. And so for me, this project is really combining several of these aspects of thinking about how prehistoric hominins are living on the landscape, how choices in our food are shaping evolutionary history, and then thinking about how we can use modern analogs from the forensic world to understand what's happening with stable isotopes. And I do all of this research here at Purdue in the Bioanth Isotope Ecology Research Lab, affectionately referred to as the Beer Lab. And so I've had a great number of students that have also helped with this research and lots of processing of samples. So how I go about asking these questions in terms of thinking about how humans and the environment are interacting is through stable isotopes. And I'm making an assumption that most of us have heard at this point in anthropology about stable isotopes, but I wanted to at least give a brief primer of thinking about what we're talking about with an isotope. And so we're looking at an element, and there's going to be same number of protons, different number of neutrons, and therefore we're going to have a difference in a heavy isotope relative to a light isotope. And so whatever your isotope of interest that you're looking at a delta value that is referred to as that in terms of nitrogen, delta N15. So you're looking at that heavy nitrogen 15 isotope relative to that light nitrogen 14 isotope. And this is all relative to a standard. And so we can apply this with various isotopes in geology, though these would be not necessarily using a delta value, but that's a whole nother ball of wax. We can look at this in water with oxygen and hydrogen. We can look at dietary isotopes, looking at carbon, nitrogen, and sulfur. And as anthropologists, archaeologists, we can then apply these to questions of thinking about where people moved on the landscape, what environment they were living in, what type of behaviors they were practicing. And so this is kind of the methodology that I broadly am using to look at these questions of early human environment interactions. And we're lucky that we can recover these biological tissues from archaeological sites, and different tissues are going to tell us different things. If we're wanting to ask questions about what was happening during the childhood period, we can look at tooth enamel, because tooth enamel is not going to remodel, and so it's going to hold in that signature of those isotopes of when that dental tissue was formed. If we are looking at a question of adulthood or bone, we can look at kind of that last decade or so of life, depending on the remodeling rate of the particular bone in question that you're analyzing. But it's homogenizing, many years of then that dietary information. First is if we were to look at someone's hair versus their fingernails, which is going to track a much shorter, more recent period of time closer to their death, where hair is going to grow at about a centimeter a month or so, and your fingernail clippings are going to represent about 5 to 6 months prior to when the end of your clippings. So will represent about 5 to 6 months prior in life. And so these different biological tissues are essentially snapshots of what was happening in terms of the isotopes that were being incorporated into these biological tissues. So we can use these different tissues to ask different anthropological questions. In terms of being interested in human dietary reconstruction, I kind of like to start thinking about this in terms of we can get at the paleo diet of a population from many different avenues of research. We can look at faunal material, but that's going to be influenced by what's preserved, what's recovered, and often you might have over under representation of particular species for various reasons. Floral species, again, it's what's preserved and recovered and what is identifiable. We can look at artifact assemblages, again, being impacted by what is preserved. We can look at paleoenvironmental questions in terms of exchange and mobility. So we can get at all these different lines of evidence. And with stable isotopes, you're not still going to get at that exact diet because we're looking at that averaging of that biological tissue that you're looking at over multiple years. So none of these single avenues are going to be perfect, but hopefully you can use them in combination to be able to get at this question of paleo diet to see what actually is a group or an individual consuming. But I think that we need to broaden that question to think more about what is available. So it's not just about what can we reconstruct from the archaeological record, but actually what was available on that paleo menu, because sometimes there are going to be food items that are not preserved in the archaeological record. As well as when we think about how food is processed, we need to think about how that is going to impact isotope values. So is there some sort of cultural food processing that might, in a sense, this is from a great paper from Hopman et al. 2020. They were looking at Inuit practices of food preparation, and immediately in the storing and the drying of meat, they observed larval eggs. So fly are coming and laying their eggs, you get those initial maggots, and that was not prevented from these traditional practices of food drying. As well as we know that there's a ton of different ways individuals are going to be able to prepare food through boiling, through frying, through roasting, and all of that is going to have potentially that impact to those isotope values. So when we're thinking about the paleo menu as that kind of broader umbrella of thinking about dietary reconstruction, there are a lot of different inputs that we would need to account for to think about what's actually happening on the landscape and what is available to these early hominins. And so what really should we consider for the Neanderthal paleo menu is kind of where this question got my colleagues and I thinking about this. And really this story starts with this personal anecdote of I was a recent PhD. I was at UC San Diego as a visiting scholar for a year teaching classes, and John Speth came to San Diego to give this talk on a paper that hadn't yet been published in 2017. It was in production at Paleoanthropology, and it was this idea that the high nitrogen values in Neanderthals could be explained by the consumption of putrid meat. And at the time, I was doing I had an application in for a postdoc at University of Tennessee, Knoxville, at the world famous Forensic Anthropology Center that is affectionately known as the Body Farm, where they do intentional donated human decomposition research. And so after the talk, I was so excited because everything that John had talked about, I said, John, I could do this. I could test this. If I get this postdoc, we could actually look at this. And so this all culminated from this very first presentation that John and I talked about this project back in early 2017 in this paper that just came out this summer and is the title of my talk today, Neanderthals, Hypercarnivores, and Maggots, Insights from Stable Isotopes. And so really, as I go through this talk, I want to start with this image of these wonderful maggots. I actually took this picture of a maggot mass in January down in Texas at another donated human decomposition facility at Texas State in San Marcos. And what I find interesting is this maggot mass, which I want to kind of keep in your mind as I'm going through the talk, this is a very easily collectible, large unit of food, of nutrients that can be collected on the landscape. And even in January, this photo was actually taken after there had been freezing temperatures in Texas. This is a very live, active colony of a maggot mass that is underneath a decomposing carcass. And so I think that it's important to think about when I'm talking about maggots throughout this talk, that this is kind of really truly the image that I want you to hold on to. And now I'm going to convince you why John, Julie, and I think that this is an important paleo menu item to be considered when we're talking about early hominin diets. So the big question with Neanderthals and their nitrogen values ever since the 90s, when these first ecological food webs came out with the stable isotope values, is why are Neanderthals always plotting at the top of food webs? I just gave a couple of different examples here from sites in France, Belgium, Croatia, but all of these, kind of this pattern is consistent at all sites that Neanderthal isotope work has been done at, where they are always plotting at the top of the food web. They're usually above the carnivores, or at least with the carnivores, as you can see with the French site. And so this is a question, because in the end, when we're thinking about this, and so when we're doing this with the Neanderthal values, we can look at this through the bone collagen values that are preserved at a site. But to ask this question of the paleo menu, we went with different analog tissue, looking at muscle tissue and maggots, which are not going to be preserved in the archaeological record. So where this idea comes from of looking at the muscle tissue and the maggots is that from this original 2017 paper that John Speth did, he collected ethnohistoric evidence from around the world. And just my favorite quote I want to read is, "To my horror, my companions scooped out handfuls of crawling things and ate them with evident relish. I criticized their taste, but they laughed at me and said, 'Not illogically, you yourself like caribou meat. And what are these maggots but live caribou meat? They taste just the same as the meat and are refreshing to the mouth.'" And so when we're talking about various groups that are following these indigenous foodways, we're talking about definitely that a delicacy comes from this processing of meat that is in a what we would consider to be a putrid state of decomposition. Well, we have to remember that hominins are primates with largely vegetarian-derived digestive and metabolic systems, and we are not specialized flesh eaters. While it's possible for humans to subsist on a very carnivorous diet, in fact, many traditional northern hunter-gatherers, such as the Inuit, subsisted mostly on animal foods. Hominins simply cannot tolerate the high levels of protein consumption that large predators can. A modern human weighing about 80 kilograms, the estimated body weight for a robust, cold-adapted Neanderthal male, cannot consume more than 300 grams of protein per day without serious health consequences. At sustained intakes above the level for as little as 2 weeks, individuals become vulnerable to a debilitating and potentially lethal condition known to early explorers as rabbit starvation. So this is a quote again from the ethnohistoric record. "We tried the meat of horse, colt, and mules, all of which were in a starved condition. We consumed the enormous amount of from 5 to 6 pounds of this meat per man daily, but continued to grow weak and thin until at the expiration of 12 days, we were able to perform but little labor and were continually starving for fat meat." So this this quote talking about this very lean meat, these animals that were in this mode of starvation, they were not getting the complete nutrients that they needed to sustain life of having protein and fat and carbohydrates together. So in modern medical terms, the consumption of such high levels of protein exceeds the capacity of the liver to upregulate enzymes involved in the synthesis of urea, with the result that the liver can no longer effectively deaminize the amino acids. And this leads in turn to the buildup of ammonia and excess amino acids in the blood. And so in contrast, when we think about, like, say, a large African lion, a lion can readily subsist on protein intakes that would probably prove lethal to a human in a matter of a few weeks. So hypercarnivorous species, such as a lion, such as a hyena, such as a wolf, they on average are going to consume anywhere from double to 4.5 times more protein per kilogram of body weight than the absolute maximum a late Pleistocene hominin would be able to tolerate. Thus, these northern hunter-gatherers, when we think of them as being carnivorous, they're carnivorous only in the sense that they relied heavily on animal foods, but most of what they ate was fat, so a fatty meat, not pure lean meat muscle tissue. And so if humans are limited to 300 grams of protein per day, then the remainder of their daily energetic needs cannot be met by just eating more animal protein. That would result in that rabbit starvation. The energy deficit instead must be made up with a non-protein source such as animal fat, plant oils, carbohydrates, or some combination of these foods. And so when we think about this in terms of this processing, I think it's important to note that putrid and pathogenic are not synonymous with one another. When we think about words like fermented, spoiled, putrid, rotten, none of those terms are actually going to mean that they are rancid or potentially harmful to humans. And so this question of, well, how do we test this question of this fermented meat comes into play? Well, judging by the ethnohistoric record, this backup reserves of animal foods, either as packets of processed meats and fat or as partial or complete carcasses, would have readily attracted flies while they were being processed. And over the use life of whatever that reserve is that was being stored, the contents almost inevitably would begin to putrefy and become infested with these maggots. And so the gap between kill and final consumption, even for animals simply cached in rock piles on the surface, often would span weeks, months, different seasons. So a fall kill not being utilized until the winter or the following spring, or these caches even being utilized a complete year later. And so, contrary to expectations that early Arctic explorers repeatedly noted that whole unskinned caribou reindeer carcasses were killed in the winter, even in very low temperatures at negative 43 degrees Celsius, they would begin to putrefy far more quickly than similar carcasses that were killed in the summer. And though their explanations for this are counterintuitive to the phenomenon, the answer seems to lie in this remarkable insulating properties of that deer's heavy winter coat, which would effectively keep the animal's body heat trapped within the carcass, and most especially in those cached without being gutted first. And so that decomposition process, even in the cold, is actually beginning to start early. Importantly to note, when we're thinking about what Indigenous peoples are thinking of as a delicacy, Indigenous people almost universally viewed thoroughly putrefied, maggot-infested animal foods as a highly desirable fare and not starvation rations. And so, in fact, many such peoples would routinely and often intentionally allow animal foods to decompose to the point where they were crawling with maggots, in some cases even beginning to liquify and inevitably emit a stench that was so overpowering that early European explorers, fur trappers, and missionaries were sickened by it. And yet these foods were viewed as good to eat. Even as a delicacy, when asked how they could tolerate the nauseating stench, they simply would respond, "We don't eat the smell." And so while our Western sensibilities might abhor the thought of maggot-infested foods, one even finds the vestiges of larval consumption in Europe with the delicacy of casa marzu, a traditional Sardinian cheese replete with the larvae of cheese flies. So this lovely picture here shows you this image of this maggot cheese. It is a delicacy. So northern foragers not only routinely consume maggots right along with the meat and fat, they also gathered them up separately from beneath drying racks and on-site wooden storage platforms. Another ethnographic quote. The drying is done very carelessly. The meat or fish is seldom protected from the rain. It becomes soft and so full of maggots that often they can be gathered by the handful in the ground below. So maggots are also collected in the spring from beneath the hides of a recently killed caribou and reindeer meat. Another quote, Inuit delight in eating the large spring maggots of the reindeer warble flies. When reindeer are killed, the hide is skinned back and the warbles are exposed to the underside. They are fat and salty, a spring treat. So, what we're talking about here is that while the ethnohistoric accounts show that both living and dead maggots are important components of northern forager diets at almost all times of the year, no data or no one has really considered the possibility that fly larvae raised on a substrate of putrefying animal tissues may have contributed to the high nitrogen values seen in late Pleistocene hominids. Fly larvae are fat-rich, nutrient-dense, ubiquitous, and easily procured insect resource, and both Neanderthals and anatomically modern humans, much like recent foragers, would have benefited from taking full advantage of them. And so, what is happening with the nitrogen? Why are we interested in this stable nitrogen isotope during putrefaction? Well, as you have a carcass in some state decomposing, what we have is that the lighter isotope of the nitrogen, the N14, is going to react faster in these chemical reactions, releasing these volatile compounds. And so you'll have a retention of that heavier nitrogen 15 within the tissue, while that nitrogen 14 is more preferentially becoming things like ammonia, cadaverine, and putrescine. And so when we look at so through time on the x-axis, we have that number of days, and on the y-axis, we have our nitrogen values. We have some carcass that's going to start out with some ratio of the N15 to N14. That decomposition process will start. And then after time, you're going to start to have more of that N15 relative to N14 within the muscle tissue. And as that decomposition process continues, you still have that preferential release of those lighter isotopes. And so over time, you get higher and higher nitrogen values. This is kind of the process by which we're thinking about how is nitrogen cycling through this putrefaction process. Well, why did I go to the University of Tennessee to do this research? Well, in 1981, the forensic anthropology was established by Bill Bass to accept donors of human bodies that were essentially donated to science to do human decomposition research. To date, there's about 2,000 or so donors that have, since the 80s, been donated to the facility. And at any one time, there's about 200 donors or so out at the anthropology research facility, pictured here in its current kind of landscape with the orange outline. It's around the medical school parking lot at the University of Tennessee. And at the facility, they curate tissues of hair, nails, bones, and teeth that can be studied. But we know a lot of things from looking at these biological tissues from a forensic anthropology perspective. Forensic anthropologists are most often asked to consult on questions of who an individual was, so creating a biological profile, what trauma occurred around their time of death, or the PMI, the postmortem interval, how long has someone been dead. We know that a variety of isotopes can contribute information to who someone was by looking at these snapshots of different tissues. I don't have much hope for isotopes helping out with trauma analysis, but when I proposed this project at University of Tennessee, one of the kind of forensic anthropology questions was, could isotopes contribute to this postmortem interval estimate? Is this a method that could be pursued by forensic anthropologists? And so over the period of 2 years in 2018 and 2019, we placed donors in the winter in January, and really trial one in 2018, we placed some of the donors, 10 donors each in January, and then 10 donors in the summer in July. And then the next year, we tried to place donors at various times in the winter as well as throughout the spring. Through this time, we collected 389 samples across 3 different species of flies to collect their maggots that were associated with the bodies during decomposition. So how did this work? Well, we also collected the muscle tissue that was associated, that we only collected the muscle tissue while the muscle was identifiable muscle tissue. And so here's just an average individual. If we look at the days on the x-axis, the nitrogen on the y-axis, what we're looking at is the big delta value. So we're looking at values where we took whatever day the sample was collected and we're minusing the original starting muscle tissue value. So we're getting this relative change. So this normalizes then what the pattern of these big delta values are through all the individuals. It doesn't matter what their starting nitrogen value was because we're just looking at this offset between when the muscle was collected and what was that muscle at its starting point. And this is the kind of similar pattern that we get for the individuals in terms of nitrogen decomposition of the muscle tissue. So it did in fact prove what John suspected with the nitrogen, that through muscle decomposition, through this putrefaction process, the nitrogen values do increase over time. And so this came out in a paper in forensic anthropology, looking at this change in the muscle tissue with individuals being placed in the summer and the winter. And in the winter case, there's longer time for decomposition. And we do see this more dramatic rise in nitrogen values because of the fact that there is a longer period of decomposition where the muscle is still present in the winter, where in the summer we see the increase in the value, but it's a much smaller increase because decomposition in Tennessee happens much faster and the muscle tissue is gone after about an average of 5 to 7 days. And so this period of rising, we are talking about in some cases as much as 2 to 3 per mil in change, which would actually indicate a trophic level difference in the nitrogen values. Additionally, we collected the fly larva, which came out in this initial paper of talking about the fly larva in terms of a post-mortem interval estimate. And what was interesting is that the larvae that were collected on the individuals that were placed in the winter were significantly different from those individuals that were placed in the summer. And so we could kind of create this sectioning point of estimating if an individual died, did they die in the winter or in the summer? And we were, correct. I think it was about 85-ish% of the time or so. And so we took this data and kind of looked at, here in this graph, our x-axis is the ADD. ADD is the adjusted degree day. This is accumulated degree day. And this is an adjusted value that I have done based on donors at Tennessee. They don't always come in the exact day that they die. So sometimes they go into a freezer for various reasons at the funeral home initially. So all of these have accounted for all of the temperature that affected the individual between their date of death and their actual placement at Tennessee. And then the accumulated degree day is this measure of essentially how much temperature was affecting the body. So this normalizes not just the number of days, but this takes into account that you can compare individuals in the winter and the summer together, even though you're in these different environmental periods, because we're taking temperature as this measure of decomposition. And so we're looking at temperature as kind of this control as a way of how much temperature has a body been exposed to through this decomposition process. And so here we can see this nice pattern of this increase over time in nitrogen values from the initial button muscle tissue to then the blowfly larva, which they are an earlier arriving to decomposition species. And then the cheese skippers, which kind of then come next. And then eventually you get those late arrival species of the black soldier flies. And we can see this difference distinctly between those black soldier flies from individuals that were placed in the spring and summer versus those that were placed in the winter. But we see this continuous pattern of the increase in nitrogen values. So then what we did in this paper is we looked at how did these maggot values compare to the fauna that has been published. Now, to date, the highest nitrogen value that has been published in terms of these Pleistocene sites with herbivores is 11.2 per mil. And so by the way of contrast to this, we have about 75% of the fly larvae that were raised on the putrid tissue in our study have even higher values. And those fly larvae that have lower values were either the early arriving species or they were the late arriving species, but they were collected from donors in the spring and the summer. And so when we went to compare the fly species to all of these different herbivore species at different sites, one of the questions was kind of thinking through, was there a difference at these different sites? We're combining just kind of all bovids, all cervids, all the different species of herbivores, carnivores that have been published, and is there a difference in these sites? And as you can see in this graph, yes, there is a slight difference between sites of maybe a per mil here, a per mil there, but that really when you look at these averages of sites across different geopolitical boundaries of different countries, not that that would have been, of any consequence to the Neanderthals and anatomically modern humans, but by just looking at these different regions of Europe, there isn't really a significant difference in the general isotope pattern of these values for these herbivore and carnivore species. So we felt very good about just kind of combining this in terms of our analysis across different sites. And so when we look at this, as if we were to take the fly larva values, and I have on the graph the initial muscle tissue substrate, so that's essentially the initial starting value of all of the human donors, which range from 7 to 9.2 per mil. If we were to take that value and we were to adjust that initial muscle tissue substrate down to that herbivore value, that initial Pleistocene fauna value for the herbivores, then the fly larva average of the delta N15 values would shift from an average of about 17.5 per mil to 14.6 per mil, which still exceeds any value of terrestrial or aquatic fauna that would be available to Neanderthals and anatomically modern humans. And so while we don't have a time machine, we can't go back and see were they actually putrefying meat and eating meat animal foods that was in a putrid state associated with fly larvae. We are proposing that it is reasonable with this higher value that this could be an input that is raising that nitrogen value of Neanderthals higher than we would expect for that trophic level position of a hominin. And this is based on this idea, not just from the ethnohistoric evidence that we have from around the world that fly larvae are seen as this delicacy, but the fact that fly larvae in this big maggot mass would be unavoidable. So hominins would have had to contend with this the minute that they start storing food, as there was not going to be a way to store food to prevent flies from coming and landing and laying their eggs. These maggot masses are also very easily collectible, as well as they're nutritionally beneficial. So you're having that, going from having that lean meat of an animal that you're storing, and you're now adding in fly larvae, which is going to be this fat-rich nutrient. So you're going to get a more complete food where you're getting the protein and that fatty maggot. And so I think the kind of conclusion of this, while we can't say for sure, yes, Neanderthals were eating maggots, I think that really the important takeaway is that we should start thinking about the fact that Neanderthals aren't special, but really that all of Homo are special. I love this figure that was in a paper by my PhD advisor, Margaret Schoeninger, from 2014. And what she plotted here was looking at the European Neanderthals and their ecological food web in comparison to the Great Basin foragers that she had worked on some material back in the 90s. And she plotted this showing essentially that where the Homo modern humans of these Great Basin foragers are plotting, they're plotting also above the Great Basin canids, above those carnivore species. They are about approximately the same plotting of where the Neanderthals. And in fact, if I go back to this graph, the average of the Neanderthals and the anatomically modern humans is exactly the same for the published literature of individuals at various sites. And so while we have this narrative that Neanderthals are special and unique for plotting high in these nitrogen values, really it's not that Neanderthals are just special, it's that all of Homo is special. And so I think that what's important to kind of take away is that it's that paleo menu that makes hominins special. It's the fact that they're doing food storage that could become infested with maggots, and they're cooking and they're doing these different food processing ways, cultural modification to the food that other animals are not doing. We know this is another 2017 great papers in isotopes. Royer et al. This was a paper published in AJPA. And they looked at this experimental study of what happens to the nitrogen or to the isotope values when you do different cooking practices to both mammal meat as well as fish. And they see these changes in nitrogen, oxygen, carbon values for different species, different cooking methods. So we do know that cooking is going to impact those isotope values. And so when we're thinking about how are we reconstructing a paleo diet, we really need to readjust our thinking to this paleo menu. How are people actually consuming the foods that they're eating? And so with that, I think I'm just a little bit early. So I hope that there are some questions, but I do want to take the time to acknowledge, first and foremost, the individuals who take the greatest gift of life and they donate their bodies to the Forensic Anthropology Center so that research like this can be done. Specifically thanks to my postdoc advisor, Donnie Stedman, and then others in the isotope lab, Anya and Anthony, as well as Hayden McKee and other undergrad volunteers that helped at Tennessee with this research. And all of this was funded through University of Tennessee, Knoxville, Haslam Postdoctoral Fellowship. So with that, I will be happy to take any questions. I don't know. I feel like that's not a fun slide to land on. So I'm just going to leave it on that back slide. And then we'll be happy to answer any questions.
Wow. Thank you, Melanie, for sharing that research with us. That was really, really interesting. If anyone in the audience does have a question, please pop it in the chat for me or use the raised hand signal and I'll call on you. I've got one to begin with. Is the high nitrogen values restricted to late Pleistocene Eurasia, or do we see it earlier?
Do we see it earlier? I was going to say we definitely see it later. I think that the nitrogen is a tricky thing going back because until recently, with the great work of Tina Luddick and Jennifer, oh crap, what's Jennifer's last name? Jennifer, who's wonderful. You haven't been able to get nitrogen out of anything but bone collagen. And so the limit to going back in time for these questions has been the preservation of the organic material. So when you're prepping to get nitrogen, we've traditionally thought of nitrogen is going to be in that bone collagen. And most of the time when we're looking at isotope values at earlier hominins, we're looking at the enamel carbonate because the organic material hasn't preserved. And so we're restricted to what's in the dental tissue. Well, recently, Tina and Jennifer were able to develop a method with others to extract nitrogen from tooth enamel. And so it is a very exciting time. They've started to look at nitrogen. I think the earliest one is Schwarzkranz that just came out maybe in the last year. And so I think that we will start to get a picture of when these nitrogen values increase. as we start applying this to tooth enamel and going down this question. And then honestly, I'll take heat for this probably later, but I think that this is going to be potentially a way that we can start looking at the when we see that nitrogen outside of that trophic level expectation. So when it is higher than where it should be, if you're just eating, say, a deer on the landscape, that I think is when we start saying, hominins are storing food, they're cooking food, that we are not just another ecological species. Because you have to remember, and actually I have extra slides. When we think about like what happens with isotopes and mixing models and understanding where, what we're doing with our food, all of these methods are developed in ecology based on non-human species. They're eating relatively few, dietary foods. They're not eating as wide a variety, and none of them are cooking, none of them are storing food. And so I think that when you have a method that is built on looking at other non-human animals, and then you place humans into that method, we have to start thinking, wait a minute, but what makes humans special? Well, we cook, we store. And so can we identify that by how early in time do we see an increase in nitrogen values that is out of sync with where we would expect that to be for just any non-human animal? When is that trophic position higher than expected to be?
Brilliant. Thank you. We've got Walter with his hand up. Walter, would you like to ask a question?
Sure. Thank you for calling on me. That was a very interesting lecture, Dr. Beasley. You combined biochemistry and paleoanthropology. So I'm interested in knowing whether at these sites there was also evidence of collected plant matter. I'm wondering where the Neanderthals got their vitamin sources. And also, were these decomposition sites near a water source? Because life needs water to be.
That's a great question, and I will be honest, I did not look specifically at locations of where all of these sites and where all of the fauna have been collected, because that I felt that's kind of outside of the scope of what we're
Sure.
talking in terms of the modeling of the isotopes. But like you say, you have to have water. I feel like all hominins are going to be in sites where there's water close by and accessible. In terms of the plant foods, Amanda Henry, I can't think of any other author on the top of my head, but a lot of Amanda Henry's work has looked and focused on a lot of the plants that have been recovered from dental calculus. To my knowledge, I don't think that we've looked at the isotopes of enough particular plant parts to think about what is happening. I look at this one, I think maybe it's not on this graph. No, it's on a different graph that Margaret has. She's looked at the roots of cattails, which I know are a very high nitrogen. They have very high nitrogen values. So I think that there's a lot of work to be done in thinking about what different plant parts have different nitrogen values, and then what cooking preparations would also impact their specifically the nitrogen values and just isotope values in general. There's definitely not been enough experimental work done in those areas. So while we know that definitely Neanderthals, we know they were eating plants. We have that directly there in that dental calculus from Amanda Henry's great work. But in terms of connecting that then specifically with isotope values, I am not versed in any literature that is specifically tied those pieces together.
Good enough. Thank you.
Yeah.
Thank you, Walter. We've also got some comments in the chat. Gabrielle says, Melanie, congratulations on a cool study.
Thanks, Gabrielle.
From Chris Stringer, we should consider how they cache the carcasses as well to avoid attentions of competitors like hyena who would be attracted to the smell.
It's absolutely. So, in fact, I was just with a colleague, Andrew Somerville at Iowa State, and we were talking specifically about how we can design a study to do some experimental feeding, so we know what the species was eating, and then be able to go from that to then actually caching and doing experimental archaeology of we take those carcasses and we intentionally store them following native practices and see what happens. So yes, that is, I hope, on the landscape for me, on the landscape for others. Karen Hardy up in Scotland, she and I have been discussions as well with John Speth of doing another step of looking at some of these intentional food caching practices. So I think that, hopefully this will inspire lots of, master's projects, PhDs, other research. There is a ton of room in the experimental archaeology world to be able to really try to get at these questions. So absolutely 100% agree. This was opportunistically at Tennessee, a way to look at the underlying what's happening to the nitrogen values, but this is definitely not experimentally a great design study for thinking about how the food preparation and storage was exactly happening.
Fabulous. Yeah, it sounds like there's a lot more research to be done.
And if anyone wants to do anything, happy to chat, happy to share my experience.
Yeah. Next question from Lydia, who's asked the question that's on everyone's mind, I think. Have you tried any maggots yourself?
No. Okay. So yes and no. I have not tried maggots fed on decomposing human tissue. I felt like there was a line there and that was not going to happen. I have had other maggots in other situations because you actually can buy black soldier fly maggots. Most of the preparations have basically been freeze dried. They're just like crunchy and almost taste like nothing. So I haven't had them like fresh and wiggly live on the caribou meat yet, but I'm hoping with future studies I'll be able to do that. There are actually companies here in North America that have invested in looking at black soldier flies as a protein source, as an alternative, more ecologically friendly protein source. And they're doing things like making like a flour. So basically growing them on different substrates, and then I guess they pound them, they dry them in some way and then pound them into a flour. So I think that, there's a lot of interest in these more ecologically friendly food sources, and insects, grasshoppers, fly larvae, all of these are going to be on the landscape of the future to feed our ever-growing population in the world.
Yeah, I think that's a good idea. And you should come back and let us know once you have tried them from caribou meat. I'm very curious. Lydia also asks, do we know roughly the proportionate reliance on meat and plant ideals between Neanderthals and ancient and modern humans, and the extent of variation with the place and time of year, etc.?
I'm going to go with that depends on who you talk to. There are ways of modeling these with these mixing models. Mixing models originally came out. Paul Koch did a lot of that work with Seth Newsom, and they did some great mixing models. And you kind of get this, Bayesian approach of thinking about like what are those inputs, and then you talk about like percentages of what potentially was going in. I am anti-mixing model. And this is kind of where kind of the arc of, I think, my research and contributions to the world are going, is that I think we need to look at Homo specifically as a unique species that we can't apply some of these ecological models to, because we do cook and we store food. And so I think until we have a better idea of that paleo menu issue, I think it's going to be really hard to definitively talk about like percentages and amounts, but people do it. People absolutely apply these fancy statistical Bayesian models, but none of them, to my knowledge, take into account anything with invisible food items, so things that are not being collected archaeologically, as well as none of them take into account what cooking, storing, would do to these isotope values. And so I think, in my opinion, it's one of those you have this flawed model, and you're going to get an answer because you put data into it, and so you're going to get an answer that breaks down into these amounts and these percentages. And of course, we can go back to the fact that evolutionarily, we're primates. There's going to be a limit. We're not a carnivorous species, so we're not going to be able to eat the amount of protein, as I already talked about, that a lion or a hyena is going to be able to consume. We kind of hit a maximum ceiling of that amount of protein we can consume. You can't just look at it as a percentage, and say, oh, well, I'll eat more protein and then I'll just eat a different percentage and I'll keep within this, that amount will change. It doesn't really work like that. And so John Speth really kind of pounded that into my head when we were having all of these conversations of a lot of the nutritional literature will present things as a percent. And so it gives this false impression that, well, if you just keep adjusting the percent correctly, then you can eat whatever you want. And he's like, but no, there's actual metabolic maximums that you can't go beyond. And then it doesn't kind of change. It doesn't matter what additional food you're inputting into, because there is that maximum amount. So I think it's a great question. Definitely the reviewers were pushing me in that direction when I was writing this paper, and I was digging in my heels of like, no, we don't have enough information. I'm a very conservative interpretations, though I'm throwing out maggots, and this might seem wild to most people. I'm still at we need to know a lot more. We need to like do a lot of these experimental things before we can definitively say, yes, we can create a model that will account for all of these weird things that humans do in terms of food processing. Fermentation. If anyone wants to talk to me about fermentation, I think fermentation has a lot of things, like fermentation of plants, very interesting as well.
Yeah, excellent. We've got a lot of thank yous from
Thank you all.
Chris, Bruce, Tony, and another question from Jim Bob, who asks, would there be any difference in which carcass elements would be ideal for maggot harvesting? I.e., could you make the use of fatty meat now and then reap the benefits of lean meat through maggots later on?
I think the short answer is absolutely yes. I think that they would go for those fatty parts initially, and then they're storing other parts. Often, a lot of the times, like the rump and the roast, which we think of now is like, right, Sunday roast in the UK. That's an amazing thing. It's an amazing tradition. Yeah, but prehistorically, people would have been feeding those to the dogs. Like that muscle tissue is not what was sought after, that we know of from the ethnohistoric record. And so I think that looking at different parts and seeing what the larva attracted to, what happens, I think that that's important. It's very hard to design ideal studies that you can go after that, as I found at Tennessee, because, one of the inherent flaws of this when you're doing decomposition research is the minute you collect a sample, which is so important to understand the change in the pattern, you're altering that decomposition environment. You're inputting like you have to cut in and you have to get a sample, and then you have to somehow close that up and then come back a few days later and cut in again. And so I think it's hard to design those in an ideal world. But I think, yes, those things are important. If I were to go back and do it again, I would collect the soil. See that lovely maggot mass there, like that soil that it's living in. I didn't collect any of that. That soupy, kind of nutrient dense, rich soil is where a lot of the fly larvae, after there's muscle tissue that isn't collectible anymore, after there aren't organs, there aren't skin, there's nothing, they're still coming and growing these maggot masses in the soil. And that's just all that decomp fluid. And I should have collected it and I didn't. And yeah, so there's definitely a lot to be done to understand what's happening specifically in terms of the maggots with the nitrogen, and then thinking about different cuts of meat as it's stored. But I think you're going to be able to do the different cuts of meat, the fatty parts, the non-fatty parts. You're going to be able to look at that better when you're doing an experiment designed in terms of butchering practices and storage. So yeah, like I said, there is a ton of research that is fit for student projects, honors theses, PhDs. There's a ton of things that can be explored in this area.
Excellent. Thanks, Melanie. Thanks, Jim Bob. Looks like we've got time for just one more question. We're nearly at the hour now. Walter, do you have a follow-up?
Yes, this is more of a comment and question combined together. I'm interested in the attraction to the smell of putrefied meats. So it seems to me that uh, because of the exigencies that Neanderthals had, it may have been a cultural attribute to approach the awful smell of putrefying meats. And are there any people today that like that? Because they know that if they approach something that smells awful, there is nutrition there. We, on the other hand, we moderns are turned off by putrefied meat. We have refrigeration. We don't grow things anymore. It gets transported. So we think that if something smells awful, it must be decaying. We're not going to eat it. We're going to get sick. Whereas the Neanderthals and maybe other indigenous people who are alive today do just the opposite. So interesting how you've combined, you know, biochemistry and biology and paleoanthropology with cultural anthropology in this topic.
And all of that credit I must give to John Speth. He is an amazing colleague and just finds all of these beautiful ethnohistoric quotes. His knowledge is deep and wide and varied in terms of the ethnohistoric record. And he really pulled this idea together originally. All the credit goes to him from that 2017 paper. And I think he addresses this best by thinking about the psychology literature. So he actually pulled in psychology as well. And the disgust response does not kind of set in humans until about the age of 5 to 7. So if you're a little kid and you're growing up and you're seeing mom and dad eat this delicacy of this putrid, dish, and mom and dad are saying yum, and you're like 2 or 3, and you're like, oh, mom and dad say that's great. That's okay, that's delicious. That is, you're never going to get that disgust response. And we could look at cultures around the world and think about that. So my boyfriend last night had kimchi for the first time. I love kimchi. I live with my sister currently. She's living with me, and she hates it when I eat kimchi because it just absolutely smells up the whole house. And she's like, "Oh God, why are you opening that up?" And I'm like, "What?" And I just sit there and I eat it right out of the jar. I love it so much. And so last night I brought over some kimchi to my boyfriend's, and I was like, "Hey, you've never had this? Like, why don't you try this?" And he's like, "Oh, that's a very strong flavor. Like, I'm gonna have to get used to that." Well, we then went out to the pub for pub trivia, and we came home, and he immediately was like, oh my God, I see what your sister means. Because the house smelled like kimchi from hours earlier, I having opened up this jar of kimchi. And he was like, that's really pungent. And so I feel like you can have examples of this where I think kimchi's delicious. I don't know about you. Stinky cheese is another one. I grew up you cannot have a stinky enough cheese in your refrigerator. Blue cheese, camembert, all of it is amazing. And of course, other people who the minute any kind of cheese smells, they're like, oh no, I cannot eat it. So I feel like that what you're exposed to, that disgust response, what's set in, that plays a big part in this kind of Western ideal of like some stinky, stenchy food. You're either going to be abhorred by the idea, or you're going to be like, well, I like other stinky things. Let me see and try that out. But yeah, I spent the summer up in Alaska, and one of the reasons to go up there was to try to initiate some connections with Native groups and colleagues to do some of this experimental research with individuals that are still practicing these traditional food processing ways. So again, lots to be done in this area in terms of people who are still eating these types of delicacies. But thank you, Walter. Great question.
Yeah, thank you, Walter. We're going to have to call that our last question now because we are at the hour, and I'm sure we could all talk about this topic all day because it's very fascinating.
Happy to chat with anyone ever about it if you ever want to send an email.
I'm sure, if anyone's got any burning questions left over, you can shoot Melanie an email. So thank you, Melanie, for your talk, and thank you everyone for watching, and I'll let you get on with the rest of your days now.
Thank you so much for having me. Thank you everyone for attending.
Bye.